Hi guys, can you hear me?
Yes.
Nice kitty. Okay. So what is it? Monday, October 19th. We're going to be finishing chapter 7 today. We're going to be talking about carbohydrates. And the homework will be due Wednesday morning. Don't forget about that. So we started, no, we haven't started glycosidic bond yet. So we talked about, we almost finished monosaccharides. Let's just briefly go over some of the important monosaccharides to summarize our discussions. Okay.
I was having problems trying to, I was looking over the anomers, and I understand like it's axial and equatorial positions determine, how it differs, but is it once it cycles, once it becomes like the ring, is that when we determine whether it's an anomer? Once it's a ring, or once it's or when we first see it like as a line?
Yeah, the anomeric position anomeric position pertains to the ring form only.
Okay.
Yeah.
Thank you.
So in the open chain, you just have the aldehyde or a ketone. So.
Okay.
All right. So glucose family. So we're going to talk about. Let's just briefly overview the monosaccharides from glucose family. So this is a beta D glucose. So here we have why can't I? Okay. So beta D glucose, remember what beta stands for, remember what D stands for. Now this is glucose phosphate, very important derivative of glucose phosphate. This is the first step in glycolysis. This is when glucose enters a cell, right? Any kind of cell, and it's committed to undergoing glycolysis, which is basically the conversion of glucose, metabolism of glucose to produce energy. The first step is phosphorylation at position 6 here. And that's a committal step. Once it's phosphorylated, there's no way back. So glucose phosphate is a committed intermediate to glycolysis. And also, there's a second function for phosphorylation. So let's say glucose goes back and glucose goes in and out of the cell, right? So there are glucose transporters. They're called GLUTs. GLUTs. Very important family of molecules which allow glucose to go back in and out of the cell. GLUTs, glucose transporters. So once you phosphorylate, do you think the glucose can get back out of the cell?
No, it traps it.
So what traps it in the cell? Why is it trapped in the cell?
I think because now it has that charge, so it can't go through the membrane, the cell membrane.
Yep, that's right. Cell membrane, remember, is a lipophilic, right? So it does not allow the passage of charged species. So once the glucose becomes charged, it's trapped in the cell and it will undergo glycolysis. So so this is glucosamine, right? So basically instead of OH, we have NH2. This is N-acetyl glucosamine. This is an amide. Muramic acid. So muramic acid is the one is also derivative of glucosamine. Only the group R is a derivative of lactic acid shown here. and N-acetylmuramic acid, a very important component of cell membrane, sorry, of a cell wall in bacteria, right? Remember we talked about NAM and NAG, right? So this is a NAM, N-acetylmuramic acid. Okay, so it's a derivative of glucose. Then there's galactosamine. So remember, I told you glucose a good starting point for galactose. You look at carbon 4, that's an epimers carbon 4. This is down, galactose is up. So beta D galactosamine, manosamine, this is an epimers carbon 2, right? So the amino group or the hydroxy group in mannose will be up compared with glucose, where it is down. So it's a C2 epimer, this is C4 epimer, C4, this is C2. All right, deoxy sugars. So now remember these ones are L sugars, right? So you can see here, the carbon 6 position here, CH3, it's deoxy, right? So there's no hydroxy group here, but it is down. It is down, which means that it's an L sugar, and it's beta. This one is beta because OH and the CH3 are on the same side. Now, if here the CH3 and OH on the opposite sides, so it's an alpha anomer, right? Alpha anomer, beta anomer. All right. So if you oxidize carbon 6, that's glucuronate. If you oxidize carbon 1, that's gluconate. And if you form a lactone, remember lactone is a cyclic ester, right? Remember from OChem what lactone is? Lactone is a cyclic ester. And so here we're gonna use this hydroxyl group and this carboxylic group to form an ester. And it's a, anybody knows what this delta means? Why is it delta lactone? So we have alpha, beta, gamma, delta. Why is this delta? Like so. It's because this hydroxy group at the delta position is the one that forms the ester, right? So for example, if you use this hydroxy group, your lactone will be 5-membered, right? If you use that hydroxy group, it's 6-membered. If you use that one, it will be 7-membered, right? So this way you can specify which particular hydroxyl will form the ester. So this one is a delta hydroxyl. And the last one is sialic acid. We'll talk much more about sialic acid when we talk about lipids, because it's overexpressed on the surface of cells, and particularly overexpressed by cancer cells. So a lot of times what people do, they develop anti-cancer drugs in which various antibodies. So remember I told you that it's very hard to differentiate normal cells from cancer cells. But let's say if there is a cancer cell, so let's say this is a cancer cell. Cancer cell. And then there and right here we have sialic acid. Sialic acid. And it has this square shape, right? But then you have an antibody comes in that binds sialic acid antibody. And on the end of the antibody, there will be a cancer drug. Cancer drug attached to the antibody. And so, so the antibody will be attached to the drug. The antibody will bring the drug to sialic acid. Once it's recognizes sialic acid, it'll sit there until the antibody and drug will undergo cleavage, and the drug will go and kill the cancer cell. Okay. Death. The drug will bring death to the cancer cell. So this technology is known as ADC. Very has a lot of promise. A number of such drugs have been approved for that, particularly in the treatment of melanoma. So ADC stands for antibody I should write this down, guys, because I might as well just put this on a test since I'm telling you about it. Antibody drug conjugates. Right. And so the only problem with ADCs is, so they work very well, extremely, there are two problems. One is obviously the cancer cells develop resistance very quickly. And secondly is the technology is very expensive, right? So basically you have to raise an antibody against sialic acid, for example. The antibodies have been raised against other markers on top of cancer cells. But let's say you raise antibody against sialic acid, you have to attach the drug to it, which involves usually pretty sophisticated chemistry to be able to attach a drug to an antibody, right? So you have to really have good, very synthetic skills. You have to have very good synthetic skills to do that. So very challenging to make ADCs. Any questions? So this is again, it's a monosaccharide, right? Which is served as a marker on cell surface and particularly overexpressed, which means present in large amounts on the surface of cancer cells. Okay. Questions? All right. All right. So another concept we need to talk about: reducing sugars, non-reducing sugars. So reducing sugars are those that have a free anomeric carbon. If you have a free anomeric carbon, then in general, you can open this. So just think about it. If you have a free anomeric carbon, forget about this one. If you have free anomeric carbon, then this hemiacetal can very easily open to produce an aldehyde. Like so. To produce an aldehyde, can open and close very easily. Now, this is not possible once you form a glycosidic bond, once you form a bond to an alcohol. So this will not open up. Okay, but this, once it opens, this can undergo oxidation and it will take you to carboxylic acid. It will take you to carboxylic acid. Like so. And you can oxidize this with a bunch of things. You can oxidize this with copper, for example, copper 2, going to copper 1. So this is known as Fehlen test. You can also use silver, right? It's a very nice reaction because you use silver plus 1 salt, and then it when it's reduced, it turns into metallic silver. So you see beautiful silver deposit, like a silver mirror on the bottom of your flask. Has anybody done this reaction in the lab? Tollens test on aldehydes from OChem or organic chemistry? No? Nobody has seen metallic silver in the lab? All right. I guess not. All right. So so these are so the ability for the sugars to have a reducing end are used in, for example, in various gadgets to measure, for example, glucose level in the blood. So you can see here, you have a beta D glucose, and there's an enzyme which is utilized. It's called glucose oxidase. And so this enzyme can oxidize this free hydroxy group, right, to form a lactone. Free hydroxy group to form a lactone. So this hemiacetal, glucose oxidase, it utilizes oxygen, and the side product is hydrogen peroxide, which then oxidizes this dianisidine, which is almost colorless. But once it's oxidized by hydrogen peroxide in the presence of another enzyme, peroxidase, so this is glucose oxidase, this is peroxidase, it undergoes oxidation to form this orthodianisidine. And this one is bright orange. So this is utilized in detection of in portable glucose sensors, right? So it's one of the common technology to measure glucose levels in the blood in diabetics. So you can see it's based on the ability of the sugar to be reducing sugar, right? So this is hemiacetal. Okay, any questions? All right, guys, are you still there? Can you say something?
Yes.
Thank you. Just want to make sure that I'm not talking into empty Zoom space.
No, you're good.
All right. All right. So I already kind of introduced you the concept of glycosidic bond. So this bond right here, once you convert this hemiacetal into acetal and you form a bond to another type of alcohol, so this is known as a glycosidic bond. Glycosidic bond, and it's used to link sugars together. So let's, what else do we need to know about glycosidic bond? So now it's an acetal, not a hemiacetal anymore. It's an acetal, and it's much more stable, less reactive than hemiacetal. Second monomer. So basically, by introducing a glycosidic bond, you're removing the reducing end, but the second sugar can still be reducing, right? The second monosaccharide. And so disaccharides are mainly named by the organization and the linkage. So for example, two glucose molecules, they can be linked through 1,2,4 bond, is described as alpha D glucopyranosyl 1,2,4 D glucopyranose. And common name for this disaccharide is maltose. All right, so all these are words. Let's now look at structures which speak thousands of words. So here is our alpha D glucose. It's alpha, OH down, this is up. It's D because CH2OH is up, and this is hemiacetal. Now this is the second, now this is beta D glucose. Now it could be alpha, it doesn't matter. Remember, alpha and beta, they undergo into conversion in different solvents. So when it's in the hemiacetal form, it's never frozen. It can always the anomers are interconvertible. So alpha glucose and beta glucose, there's also an equilibrium between them. So this is hemiacetal and this is our alcohol. When they react with each other to form an acetal now, so you can see here, this is carbon 1 of 1 glucose, and they will form this glucose will form the acetal with carbon 4. This was carbon 4 right there. So this was carbon 1. Carbon 1 will react with carbon 4 to form the full acetal linkage, right? And this still remains the hemiacetal. So we removed, so this is now non-reducing, right? So which means it will no longer react with silver. You will not see the beautiful metallic silver, but this still remains reducing. So overall, the sugar is reducing, right? So if you take this disaccharide, and the name of this disaccharide is maltose. If you take maltose, expose it to silver, you'll still see the metallic silver. So it's because it still has a reducing end. So how we're gonna name this? So we're gonna start with the sugar that has the anomeric position, right? In which the anomeric position was utilized to make the glycosidic bond. So this one is a glycosidic bond. Glycosidic bond. All right, so we start with this sugar that used its anomeric position to form a glycosidic bond. And so we name this. So it's alpha, it's D, then we say it's a glucopyranosyl, and then we specify what kind of glycosidic bond this is. This is 1 to 4, right? So carbon 1 of this glucopyranosyl to carbon 4 of this D-glucopyranose. So this is D-glucopyranose. Does it make sense? So obviously, when you see for the first time this long name, you have no idea what's going on, but hopefully by now, every number and every letter here makes sense to you. Questions? No? So this is maltose. Another disaccharide, also reducing. Now here we're gonna link beta-D-galactopyranosyl. So galacto, remember carbon 4 in this position is inverted. So this is beta glycosidic bond, which means the anomeric position on carbon 1, the bond towards this oxygen of the glycosidic bond is up. It's up on the same side as CH2OH. So it's beta. Hold on a second. Just guys, hold on a second. Okay, I'm back. All right, so beta, right? And it's going to be linked to carbon 4 hydroxy group of a glucose, and so so it's a beta glycosidic linkage, right? Beta glycosidic linkage. So we have beta D galactopyranosyl 1 to 4 beta D glucopyranose. And you can abbreviate this, right? So abbreviate this so the name is not so long. And lactose, where do you find lactose?
In milk.
In milk, that's right. You may already notice that I will talk a little bit more about this when we talk about cellulose and starch. But so these glycosidic linkages. So if I told you about this one, which is alpha, which is maltose, which is very easily digested, and lactose, you know, is not very easily digested, right? You can already see that the alpha glycosidic linkage is more easily broken than beta glycosidic linkage, right? So to break the beta glycosidic linkage, you need a special enzyme, lactase, right? Lactase, which is expressed in babies of milk who feed on mother's breast milk, right? So they need an enzyme to break down this linkage. So in adult, once we reach the adulthood, then or even before that, many of us lose the enzyme and become lactose intolerant. So the beta glycosidic linkage is much more stable than the alpha one. Just keep that in mind, and we'll talk about the reason for that a little later. So this is sucrose. So sucrose is a combination of glucose here on the left and furanose, sorry, and fructose on the right. And so glucose is going to be glucopyranoside. So in this case, we actually, it doesn't matter which one goes first because they're both in numeric positions. You can see this is the numeric position on glucose, position 1. And this is position 2, which is where the ketone group was. This is the numeric position on fructose. Now, how do you tell which one, if it's a numeric position or not? So obviously you can convert this into the open form of fructose and find that ketone and find carbon 2, right? Alternatively, the way I do it is, let's say I look at this carbon and I see that this carbon is bonded to two oxygens, this oxygen and this oxygen. That means this carbon is a ketal. This carbon is an acetal, right? Carbon 1, and this carbon is a ketal. So which means both of these are anomeric positions. So so it doesn't matter which one I use first. So here we use fructose first. So beta D fructofuranosyl, alpha D glucopyranoside. And you can see here this kind of arrow is used, which means again one anomeric position is 2 beta, which is in the fructose, and the other anomeric position is 1 alpha, which is the one in glucose. And it doesn't matter which way you go. So you can see fructose on the left, glucose on the right, glucose on the left, fructose on the right. And this is the type of arrow you will use, which tells you that both of these are anomeric positions and you can start with either one. Any questions about this? So is this sugar reducing? Tell me that.
No,
Okay, why not?
because both of the anomeric carbons are involved in the bond.
Right. So because we don't have any more hemiacetals or hemiketals, right? So this is now a full acetal. This is a full ketal. And remember, they're much harder to break and impossible to oxidize. Right. So we use 2 reducing ends and form a glycosidic bond, which means the rest of the molecules are non-reducing parts, right? And this is another example of a non-reducing sugar. So this is two glucose residues, or 2 glucose monomers are coupled, right? So both of these are going to be alpha. You can see this one is down, away from CH2OH. This one is up, away from CH2OH. So trehalose, I don't know how to pronounce this thing. Trehalose. So this one is a disaccharide of 2 glucose molecules, both alpha. Okay. Questions? All right, let's keep going. Yep. Those question?
No, no question.
All right. Okay, so these are disaccharides. Now let's talk about polysaccharides. So sugars, monosaccharides are very important. Disaccharides are important. We talked a lot about many applications, right? So polysaccharides, important biological molecules as well. So these can be homopolysaccharides and can be heteropolysaccharides. One thing to keep in mind is how you spell this. Make sure you spell this correctly. Has 2 C's actually, very counterintuitively, has 2 C's. Saccharide, actually. I use my PhD thesis had basically dealt with saccharides, and I remember I had to correct the spelling in many places in my PhD thesis. So saccharides has 2 C's. So homopolysaccharides and heteropolysaccharides can also be linear or branched. So let's look at this and we'll come back to that slide. So these are homopolysaccharides, polysaccharides. This is unbranched, right, linear, and this is branched. This is branched, so which means that you have a glycosidic bond here, and there's another kind of glycosidic bond coming off the side of the molecule, and that will create its own branch. Now this is the same idea, only with the heteropolysaccharide, where the units are different. Here they're alternating. So in this case, it's almost like you have disaccharides as a single unit, right? Like so. Okay, another thing to keep in mind is that polysaccharides do not have a defined molecular weight. So remember that this is in contrast to proteins, right? So proteins, remember, are made on ribosomes, and the information is read from messenger RNA. So the messenger RNA serves as a template. And so each protein has a certain size based on the type of messenger RNA that brings the genetic code to the ribosome. So carbohydrates, polysaccharides are not made based on a template, and instead they actually exist in a constant state of flux, right? They're forming, they're decomposing, forming, decomposing. And there are lots of enzymes that act on polysaccharides, which allow for this to be possible. Okay. Okay, so let's talk about some homopolymers of glucose. So glycogen is one important one. So this is a storage polysaccharide in animals, right? So it's a branched homopolysaccharide of glucose. So alpha 1 to 4 linked chains. So we saw that a couple slides before. And now there are also branch points. There are branch points. So every 8 to 12 residues, they'll be in an anomeric position linked to carbon 6 of perhaps the same glucose. So the glucose that has position 4 linked to a branch, position 6 of the glucose can be linked to a branch, right? So in other words, you have something like this. This is glucose, like so. and like so. Oops. So this will be another glucose and so on and so forth. So this is position 6. This is position 4. And so you have one branch here and another branch here, right? So now molecular weight reaches several millions, and it functions as the main storage polysaccharide in animals. Now a similar one, similar idea. Plants came up with their own polysaccharide to store glucose. And so in plants, this is again a mixture of actually 2 homopolysaccharides of glucose. One is known as amylose, and one is amylopectin. So amylose is just unbranched polymer of alpha 1 to 4 link residues, and amylopectin is a branch like glycogen, but the branch points, they're actually less frequent. They occur every 24 to 30 residues. And you can see molecular weight of amylopectin is up to 200,000, up to 200 million. So here's the structures of amylose and amylopectin. So you can see here, this is the linear. So amylose is linear, 1 to 4, right? So 1 to 4. So you start, let's say, with the reducing end, and this reducing end on this side, where there will be most likely an OH. And there is a non-reducing end, which will continue on, and there will be no hemiacetal on this side. So amylose 1 to 4, and it's alpha, and amylopectin. So you can see here so let's say this is the main chain. This is the main chain. So you have 1 to 4, 1 to 4, 1 to 4. But then at some point, every 24 to 30 residues, there will be 1 to 6. 1 to 6. And then you have another branch. Then you have another branch. So.
Could you go back to the previous slide for just one moment?
Yeah. Did you have a question about it, or you're just writing something down?
I was just writing something down. Thank you.
Okay. Good?
Yes, sir.
Okay. All right, so glycogen and starch, they're insoluble due to their high molecular weight and often form granules. And so the granules contain enzymes that synthesize and degrade these polymers. So obviously they will have one reducing end, only one, right? And a whole bunch of non-reducing ends because they are branched. Not amylose, right? Amylopectin. Not. How many non-reducing ends does amylose have?
Just one.
Just one. That's right. It's the amylopectin which is branched. It's only the branched polysaccharides that will have multiple non-reducing ends. They will all have one reducing end, but if it's branched, it will have multiple non-reducing ends. If it's not branched, only one non-reducing end. And so they constantly decomposing and forming, right? So I mentioned to you last time that, for example, glycogen, So it's very quickly synthesized by glycogen synthase in the liver. So let's say we have a meal, right, with glucose in it. So glucose goes in the bloodstream, and very quickly, a lot of it is converted to glycogen in the liver to store, right? And then let's say you come to my lecture, which is very difficult, especially trying to figure this out from a person with a Russian accent who can't explain things well. So your brain consumes a lot of glucose, and then what can you do? You need more glucose, but you can't take a break, right? So you'll miss important stuff, and which will be on the test. So luckily, when you were having your meal, your liver made a lot of glycogen. Your liver made a lot of glycogen, and so your glycogen is gonna start to be hydrolyzed in the liver to produce glucose, which goes back in the blood, and the blood will carry it to the brain, and which will serve as the nutrient for the brain. And so enzymatic processes occur simultaneously in many non-reducing ends. So if it only chopped one sugar at a time, obviously this would take forever, right? So the whole system would not work. But luckily these enzymes, they're all over the place, and they actually cut the glycogen in many places at the same time. Basically, they kind of unravel these chains. So I have a question for you. It's a difficult one. You have to think about it. So why do we need to make glycogen, or why do plants need to make starch? Remember when we talked about, let's say, neurotransmitters? So we talked about these vesicles in the presynaptic neurons, right, where neurotransmitters would be stored. Let's say you have dopamine, serotonin, and acetylcholine. So when action potential arrives, the vesicle fuses with cell membrane and spills the neurotransmitter into synaptic cleft. And then once the nerve signal is finished, then they're reabsorbed back into these vesicles. So why can't glucose be stored in vesicles in, let's say, same liver, right? In the hepatocytes, in the cells of the liver. Why can't it be stored as monomeric glucose molecules? Why do we need to make glycogen?
Doesn't it throw off like the, I think it's tonicity of the solution? Like if you have so many molecules of glucose, it would draw water into the cells.
Beautiful, but it's so tonicity. So use the right term. What's the right term for it?
It would make, like, do you mean like specifically what would it make the cell?
Well, tonicity. Well, no, I think you did use the right term. Okay.
Okay, because I was thinking it would make the cells hypotonic, because it would draw water into them if they were storing a ton of glucose molecules versus storing like one glycogen molecule. Okay.
Yeah. Well, the word I was looking for is osmosis, but you said tonicity, so which is equivalent. Yeah. Okay. Yeah, that's exactly right. I think. Well, that remember. There are colligative properties and there are non-colligative properties. Remember what colligative properties are? Remember the definition of colligative properties? So why you say that, so when there's so many molecules of glucose in the vesicle, the solution of that glucose will be hypertonic, right? So
those are the properties that have to do with just how many molecules of something there are in the cell. So like freezing point, boiling point, like those things just depend on how many molecules there are, not like what each kind of molecule is. Is that right?
Beautiful. Yes, that's right. So So did everybody get that? So when you make one molecule of glycogen, right, would say 10,000 or 100,000 molecules of glucose in it, then it's just one molecule. And then if it's just one molecule, based on the fact that it's a osmosis, it's a colligative property, right? So it will not draw water into these storage cells. And so they will not burst from swelling or, you know. So it's a beautiful way to store small molecules by polymerizing them. By polymerizing them into simple large molecules and then degrading them very quickly when the monomers are required. Okay. Good job. Sounds good. All right, so cellulose, another homopolymer of glucose. So cellulose is a linear homopolysaccharide of glucose. Now here we have beta, 1 to 4 link chains. So so why is it so? So cellulose is much stronger, right? So it's not used for storage of glucose; it's used for structural properties. So it's tough and water insoluble, and is the most abundant polysaccharide in nature, obviously because of its occurrence in plants, right? And cotton is nearly pure fibrous cellulose. So this is a fragment of cellulose. So you can see here, so this is beta glycosidic linkage, right? Position 4, position 1. So you can see position 1. So this is down. Oh, wait. Not this one. This one. This one is down. This one is down. So it's a beta glycosidic linkage. So beta 1 to 4 linked glucose units. And there's a lot of possibility in this kind of structure. It's linear, a lot of possibility for hydrogen bonding. Lots of hydrogen bonding, not only within the same chain, but also between different chains. And so fibrous structure, water insolubility, so very difficult substrate, very hard to break down, right? Most animals cannot use cellulose as a fuel source because they lack the enzyme to hydrolyze beta 1 to 4 glycosidic linkages. So I want to show you something. I just need the space here. There's a phenomenon known as anomeric effect. Now you will not hear about this from a pure biochemist who would be teaching this class. Since I'm an organic chemist, I can tell you about things like that. I cannot tell you about other things that they can tell you about, but this is my turf. Anomeric effect. Anomeric effect. So let's say you have an alpha glycosidic linkage. So OH. And this is OR. Alpha. Well, it turns out that, and let me draw beta. It turns out that alpha glycosidic linkages are susceptible to this anomeric effect and beta are not. So this is beta. And so there are these electrons on the oxygen which can come in and kick out this alkoxy group. In other words, if this is a chain, let's say this is a starch or glycogen, right? You will very easily break this glycosidic bond because of this stereoelectronic effect. So an anomeric effect is actually a stereoelectronic effect. So in other words, these electrons are sterically beautifully positioned to kick out this alkoxy group, right? And you can actually form in this what's known as an oxenium ion. Oxenium ion plus the alcohol, right? Now, for the beta, this stereoelectronically, this is impossible because you have to, this is like an SN2 reaction. You have to kick from behind. And here the same thing, you have to kick from behind. But this cannot kick from behind. You would have to go all the way around. Impossible. Okay? So there's a beautiful organic chemistry explanation for why, for example, things like starch or glycogen have been selected for storage properties and very rapid formation, rapid decomposition because of the alpha linkage, because of the anomeric effect. Now, cellulose was chosen by nature because of its beta linkage and because of the strength of the glycosidic bond, because it's not subject to the anomeric effect. So this is unstable. Stable. Okay, questions? So remember anomeric effect. All right, cellulose metabolism. So obviously ruminants. So we can digest, we don't have the enzyme known as cellulase, which can break down the beta linkages, right? So this enzyme must be really developed by nature to break down these beta linkages because they're not so easy to break down. And so obviously ruminants, they live symbiotically with microorganisms in their gut, which can actually produce cellulase to digest cellulose. And termites too, right? So termites, the way they break down houses is by producing the enzyme cellulase, right? So the termites produce cellulase, which can hydrolyze the beta linkages. And cellulase is not only the bad guy, it's actually, people study this enzyme because it has a lot of promise in actually converting biomass into biofuels. If you heard of biofuels, that's what it is about. Using, for example, cellulase to break down biomass into biofuels. So with petroleum, you know that we're not going to survive on petroleum for much longer, right? So our children, grandchildren will end up without energy if we do not continue exploring other energy sources. And so biomass is a renewable energy source, and cellulase can break down the beta glycosidic linkages, so it can be used for that purpose. All right, chitin. So let's see where we stand. I have 25 minutes. Okay, I still have some time. So chitin, linear homopolysaccharides of N-acetylglucosamine. So chitin forms the exoskeleton of insects, spiders, crabs, arthropods, crustaceans, shrimps, lobsters, anything like that. So it's very similar. Again, it's a beta glycosidic linkage. That's why it's so strong. It's except for instead of glucose, it's N-acetylglucosamine, right? So instead of OH, we have the amide. So another strong polysaccharide. So that's the beautiful beetle whose armor is made out of chitin. Agar is an interesting one. We're going to talk, well, we're not going to talk about the structure of agar, but just to, it looks like this. It's kind of very interesting. Polysaccharide, it's a bit sulfated, has some charge. It has a bridge structure like this, right? So it's an interesting type of polysaccharide, and it's used in biochemical assays. For example, to use agarose solutions, so which form gels, and we'll use those when we talk about, for example, separation of DNA by electrophoresis. And this comes from a cell wall of some specific seaweeds. So it's actually agar is actually a marine polysaccharide isolated from seaweeds. All right. So in the remainder of this lecture, we'll talk a bit about some of the sugars in the extracellular matrix. So some of the important ones are these glycosaminoglycans. It's a mouthful, but try to pronounce it a few times and it should stick. Glycosaminoglycans. So these are linear polymers made out of repeating disaccharide units. So one of the one part of the disaccharide unit will be N-acetylglucosamine or galactosamine, and the other will be negatively charged, could be glucuronic acid, which can be sulfated. So this is extended and hydrated molecule, and it minimizes charge repulsion. So in other words, the reason why it's hydrated is because it minimizes charge repulsion. Actually, we're going to look at this in a moment. And you can see these glycosaminoglycans are very important. So they form meshwork with fibrous proteins to form extracellular matrix, specifically of connective tissue, and also lubrication of joints. So when something forms a connective tissue, now one distinctive feature of connective tissue, for example, is the ability to withstand pressure and not break down, right? So bones, for example, brittle, right? But for example, connective tissue is not brittle, right? It's you can press on it and it will reduce its shape, but then you reduce the pressure and it will regain the original pressure, the original volume, right? So this has to do with these negative charges. So let's look at that in a second. So these are glycosaminoglycans. You can see negative charge here. So this is hyaluronate. So because it's such an important component of joints, and chondroitin sulfate, for example, you can buy this at different pharmacies, right? And so you can see there's negative charge. So here's this N-acetylglucosamine, and the second part is glucuronic acid. Here is N-acetylgalactosamine, which is sulfated position 4. And this is glucuronic acid too, chondroitin sulfate. So if you, let's try to draw this. So let's say you have a chain like this, and you have these sulfates, negative charge, negative charge. So if you have lots of negative charges, usually these things are hydrated pretty heavily. So you will have lots of molecules of water in between these charges. And remember what water does? Water reduces the charge because water has such high dielectric constant. Remember the electric constant, right? So this is something that minimizes the interaction between charges. So water has very high dielectric constant. So the two negative charges will not repel each other so much if there is water in between them. okay? But what happens, let's say, if you squeeze this whole thing? You squeeze it, water, this will reduce its volume, the water will come out, right? Because there's not so much space upon pressure. And so obviously the negative charges will be brought together, which is not desirable, but nothing will break. Nothing will break, right? You'll just squeeze it, add some pressure to it, and then as you release the pressure, the water will come back in. Water will come back in and out, in and out. So this will allow for connective tissue, and for example, for fluid that lubricates joints, which made out of glycosaminoglycans, basically to be non-brittle, right, and allow for the bones to kind of move past each other a little bit without having something, without breaking something. So I think that's what I wanted to say about glycosaminoglycans. Chondroitin sulfate. Now there's keratin sulfate, also has a sulfate. Now, one of the most sulfated ones is heparin. Heparin has carboxylate, and it has 4 sulfates. And heparin is actually a drug which is used in medicine. Anybody knows what it's used for?
It's an anticoagulant.
Anticoagulant, that's right. So obviously, if you have a blood clot, heparin can be administered, or if you have surgery that has the potential to result in some blood clot formation, heparin is given prior to surgery. And this is the segment. You can see it's an extended form. And so this is a linear polymer, 3 to 40 kilodaltons. So it has some of the highest negative charge density among biomolecules and prevents blood clotting by activating protease. Okay, so let's see how it works. So there is a protease known as thrombin, and it participates in blood clotting formation. So there's actually a large cascade in blood clot formation utilizing a variety of different proteases, and thrombin is one of them. But there is a natural inhibitor, antithrombin, which unfortunately does not work by itself. It actually requires a molecule that can actually bridge antithrombin with thrombin, and this molecule is heparin. And so remember, heparin has negative charges, right? And whenever you see on proteins, whenever you see areas which are blue colored, like this, right? The blue color usually means, what kind of amino acids are represented by blue color? When people do modeling of proteins, these are usually amino acids which can be protonated. This usually represents the areas of positive charge. So which amino acids are these? What else? What else can be protonated? No? You forgot the amino acids? Arginine?
Histidine?
Histidine, good. All right, and so red usually, the red color is usually used for negative charge. And that will be, for example, aspartate or glutamate. All right, and so heparin, remember, with all its negative charges, will bind to the area where all these positive charges are, on this protein and on this protein, and form a very strong complex with the two, and will glue one to the other. And so thrombin by coming into close contact with thrombin, will inhibit it. All right. So now, so these are glycosaminoglycans. Now there is some summary of various polysaccharides we've just discussed. Please take a look at these. We're not going to go over this in class, but this is for you, a summary, so that you remember what we've talked about. Now, some of the remaining topics are glycoproteins. So basically, one thing to realize is that even though we've talked about proteins a lot, what we've never mentioned is that half of the proteins are actually glycoproteins. What that means is that they have carbohydrates attached to them. Okay, and the carbohydrates are mostly attached to serine, threonine, and asparagine. And the carbohydrates are actually molecules which play the key role in protein-protein recognition. When the two proteins come together, usually they expose their carbohydrate regions, those interact, and they say, okay, we match each other, so the proteins will be attracted to each other or do not come in contact. So here, as you can see, one protein. So here is serine. So for example, serine can be glycosylated, right? So a glycosidic bond can be formed. So it's glycosylated by N-acetylgalactosamine. Now this will be O-linked, which means linked through this oxygen of serine. Now here's asparagine. So you can see here, this is N-linked. This is N-linked. And so here is N-acetylglucosamine, right? And you can see here, usually these are small oligosaccharides. Remember oligosaccharides? Oligosaccharides. And so these are glycoproteins. Now you can also have glycolipids. So lipids, remember, those will be found primarily in cell membranes. And so for example, lipopolysaccharides form the membrane of various gram-negative bacteria. So remember, so those kind of go on top of the peptidoglycan layer. And so this is the general structure of lipopolysaccharides. So there are actually three components. There's lipid A, which is the conservative one. Almost all gram-negative bacteria, such as E. coli, for example, they will all have that. There's a core structure, which consists of a number of carbohydrates, which also conservative. And there's an O-specific chain, which is different for different bacteria. And actually this is variable and basically serve the function of recognizing various molecules on the surface of species or organisms which are going to be infected by these bacteria, right? So, so they call different serotypes of the same bacteria. All right. Now, proteoglycans. Now, so we talked about glycoproteins and now proteoglycans, just to make our lives harder. There is glycoproteins we just talked about, but there's proteoglycans, okay? So proteoglycans are actually quite different. So here you can see, you can tell by how the word is put together. Glycoprotein usually means a big protein which is glycosylated a little bit, right? So here is actually a glycan, which has some protein component to it. So so there are two kinds. There's syndicans and there's glypicans. So structurally they're very different. So these are found on the surface of cells. So for example, with syndican, you can see basically this will be a protein core which crosses cell membrane in one part, comes out on the other side, on the outside of the cell membrane. And then there's these glycans. You can have heparan sulfate, chondroitin sulfate. This is one type of structure known as the syndicans, and the other is glypicans. Here you can see we have fatty acids anchored to the cell membrane through the lipophilic interaction, right? And then you have the carbohydrate portion known as a GPI anchor. It stands for glycosyl phosphatidyl inositol anchor. So we'll talk about these anchors a little more when we talk about lipids. And then there's a protein portion. And the protein portion also has these glycosaminoglycans, right? So also long glycan chains. And the role of these, basically they form the main components of extracellular matrix, right? And they also play a role in recognition of, for example, growth factors. For example, when you have a fibroblast growth factor arrive into the cell, it'll before it's allowed to come into contact with receptors on the cell, it will be recognized by the glycan on the surface of the cell, and the glycan will say, "Okay, I know this guy. Let me let it through. Let me let it through," and it can signal to the cell that the cell needs to undergo cell division. Right, so it's a growth factor. Growth factors are usually signaling molecules which make cells divide. And so this is a kind of a blow-up view of some of these glycosaminoglycans, which are part of the proteoglycans. So you can see there are alternating residues. There's Na and Ns domain. So Na is non-sulfated, Ns is sulfated. So you can see there's various sugars attached to it. There's idose, unusual sugar. Now, and it's still glucose, glucuronic acids, sulfated, and so on and so forth. So just a big, just to give you a general idea of what these glycosaminoglycans look like. Obviously, I will not ask you for all these details on the test, but obviously I will want you to know the difference between glycoproteins and proteoglycans, and difference in structure and difference in functions. All right, and so how are these glycans linked to the core proteins? Okay, so they're linked primarily through an anomeric carbon of a sugar known as xylose. Xylose is a pentose. So you can see here, so there is a combination of these 4 amino acids, which is when you see serine, glycine, some other amino acid, and glycine, usually that's a segment in a core protein which would have a glycan attached to it. And so you can see here serine, the high oxygen of the serine will be glycosylated with xylose, galactose. These are different. These indicate how are they going to be glycosylated, what kind of glycosidic bonds. And then you have chondroitin sulfate, which is the glycosaminoglycan. That's going to be the large glycan, which is sulfated, and which consists of the repeating disaccharide units. All right, so the last thing I wanted to mention, I think here would be, so these proteoglycan aggregates, so some of these form very, very large molecules. We can see the molecular weights are very large. And in fact, that some of these have the size of bacterial cells. It's a single molecule, the size of a cell of a bacterial cell. They hold a lot of water, right? 1,000 times its weight. So provide lubrication. So we talked about, so now again, these are sulfated molecules, they're charged. And I told you about the water being a component of these large molecules, which will provide the compressible property, which will allow, for example, for the formation of cartilages, right? Cartilages are also a type of tissue which is designed to for the bones to not to rub against each other, right, and not to undergo any kind of, not to crack under the pressure, under physical pressure. So reduced friction, right, and load balancing. And so these large aggregates look something like this, right? So for example, here you will have this repeating disaccharide. This will be hyaluranon. I think that's how you pronounce it. Hyaluranon. This is a disaccharide in the center. And then this will be a core protein known as aggrecan. Core protein, which is going to be attached to hyaluranon through link proteins shown here. And then these are brushes. The brushes are chondroitin sulfate. So the whole thing looks like a brush. So this is the microscopy image of such a aggregate. And so basically this will form the extracellular matrix, right? So material outside the cell, strength, elasticity, physical barrier. And also keep in mind, so ECM, again, it's extracellular matrix, right? Keep in mind that extracellular matrix represents a barrier, for example, for tumor cells seeking to invade new tissues, right? So, so when a tumor cells is ready to detach from a tumor and go seek other places to metastasize to start a new growth, cancer cells have to overcome the extracellular matrix. And so they actually, many tumor cells express or secrete various enzymes which degrade extracellular matrix. And so, for example, one of them is heparinase, which is the enzyme which will break these glycosaminoglycans. All right. What else do we have? Syndicans, a couple other proteins which are worth mentioning. I think we have a picture here. So many cells interact with the extracellular matrix through the plasma membrane. So for example, there is actin filament. So you know the actin is like the cytoskeleton of the cell, right? So the cytoskeleton is directly linked to proteins known as integrins. Integrins are linked by means of fibronectin to proteoglycans, which compose the extracellular matrix. So whatever is happening outside of the cell is directly linked to the cell, into the inside of the cell. Okay, so this is cytoskeleton. You can see it directly communicates with the extracellular matrix. So it's all one big thing, right? So the cells do not exist in isolation. Cells live in a community where there's a lot of extracellular matrix and there's a lot of communication between the two. For this reason, people ask about cancer research, for example. When we have cancer cells in in culture, right? And we test new drugs by their ability to kill cancer cells in cell culture. Well, one thing we have to keep in mind is that in living cancer, in clinical cancer, the cancer cells live in together with other tissues, they live in together with extracellular matrix, they're communities, right? And the communities would respond to drugs differently than isolated cells in cell culture. And so the cell culture model is just simply not a good model for cancer drug development. It's a great model to find lots of compounds which can kill cancer cells, but the fact very few of those will actually become cancer drugs. And most of these, if they ever become cancer drugs, they will be so cytotoxic that they will be generating lots of side effects. So cancer research really requires some new model systems.
That's why there's such a big difference between in vitro and in vivo, right?
Yeah, that's right. So many journals right now, if you have data for new compounds in cell culture, many journals, top journals, they will not accept the data. You will not publish a paper unless you demonstrate that your drug works in vivo in mice. Yeah, but testing in mice is very expensive. So it's not only expensive, but also you have to jump through a lot of regulations. And to get funding approved to test drugs in mice or rats or in any other in vivo model, it's very expensive and not accessible easily. So, yeah. All right, I think we done with our lecture. Are there any questions about anything today?
I have a quick question. You mentioned the cancer cells secreting, is it heparinase? And so
Yeah.
that destroys the glycosaminoglycans. So do people with cancer, does that affect blood clotting then? Because heparin, wouldn't that make blood clot, since heparin is usually the bridge between the thrombin and the antithrombin?
Yeah, so I think we're talking about
Are those separate?
this would be in like a different location, right? Different place.
Location. Got it. Okay.
Yeah. This will be an extracellular matrix, and the one in
Versus
heparin will be in the bloodstream.
In the bloodstream. Yeah. Got it. Okay, thank you.
Yeah. Okay, guys, I'll talk to you on Wednesday. Bye.
Thank you.
Bye.